I am Nicholas Tatonetti, Associate Professor of Biomedical Informatics at Columbia University, and I spend my time thinking about how to use massive, large observational data sets to make scientific discoveries. And today we're going to be talking about something called data mining. Observations are the starting point of all discoveries. I love starting with this slide. You can see this figure over here. And this is a scanned page out of Darwin's notebook. And I love this because at the top right left corner there, he writes, "I think," and then he draws this beautiful diagram. And if you stare at this diagram, you can start to see the connections that Darwin is making between geography and phenotype that, of course, would lead to the theory of evolution. And this is not just true in biology, but in a field that I study very deeply in adverse events, drug adverse events, or pharmacovigilance. In fact, there are these two scientists, McBride and Lentz, from opposite sides of the world, were making observations of their patients. They were noticing that the pregnant women who they were prescribing thalidomide seemed to have more birth defects than other women. This was a really surprising result, and the birth defects are very severe. But these observations led to the discovery and validation of thalidomide as a potent teratogen, in fact, kicking off modern pharmacovigilance and adverse drug effect studying as we know it. The tools in how we are making observations are advancing. At first, we started with our human senses. We would go out into the world like Darwin sailing on the HMS Beagle to the Galapagos Islands and look at things and taste them and see them and write them down in our notebooks. And we started augmenting these abilities with microscopes and telescopes so that we could see farther than our eyes would naturally let us see, or hear things that we otherwise could not hear. In addition to that, we started to invent different biochemical or screening technologies, different ways to augment our senses in unnatural ways. So chemical screening, high throughput screening, microarrays, and next generation sequencing are all examples of coming up with new senses, using the discoveries that we've made to collect observations at scales that we've never seen before. So when Darwin and McBride and Lentz were going out into the world and collecting observations, they were collecting bytes or maybe kilobytes worth of data. But today's scientists are collecting petabytes worth of data with every sequencing run. It's an amount of information that's very difficult to capture in just one person's and one scientist's mind. So how do we deal with that? And in fact, it doesn't just stop there. Technology is making it easier to collect massive clinical data sets, observations at a global scale about how patients are treated, what drugs they are taking, and what those outcomes might be. In fact, over 98% of hospitals in the United States are using electronic health records, collecting observations on every patient that walks through the door. Those observations, each of them could lead to new discoveries. Just to give you an idea of the scope and the scale of the type of data that's available to observational clinical research, this is an example from just one hospital, Columbia University Medical Center, where we have over 6.5 million patients. And for these patients, we have an incredibly rich data set of prescription orders, of clinical diagnoses, of procedures and surgeries that occur, and every lab test that gets run, every time your height gets measured or your weight gets measured or blood is drawn for tests. And in fact, on top of that really rich structured data, there's also the unstructured data, the notes that are written about patients, how this patient is being cared for, what is the treatment plan, what is the prognosis for this particular patient. All of this kind of represents a particular patient and is incredibly detailed. And then if you multiply that by 6.5 million, you have this massive data set. But that's just one hospital. Across the world, there are thousands and thousands of hospitals just like ours at Columbia University. In fact, the average patient generates about 80 megabytes of data per year. And that's even accounting for the fact that most of us only go once or twice. There's a problem with using these data, though, these observational data sets that look so rich and so promising, that keeps us from making huge discoveries. And that has to do with missingness and bias. And to exemplify this, I like to tell this story about a red bird and a blue bird. So I'm here in my apartment in Manhattan, a few blocks away from Central Park. And so I often go through Central Park and I like watching all the birds. And I've noticed that there are red birds and blue birds in this park. And so we're curious. So we want to go out there and figure out what's the relative population of red birds to blue birds in Central Park. Seems like a fairly reasonable question. So we grab a notebook and we walk to Central Park and we take a hike. And every time we see a blue bird, we make a mark on the right. And every time we see a red bird, we make a mark on the left. Then we get back to the apartment, to our home laboratory, and we analyze those results. And what we observed was that we observed 5 times as many blue birds as red birds in Central Park. Now, what does this tell you about the question that we asked? What is the proportion of the population of blue birds to red birds? Absolutely nothing, because now I'm going to tell you something else, which is that blue birds, you might be able to tell from this graphic, are really friendly birds. When they hear people come into Central Park, they know they drop food all over the place. They're super excited to go chirp at them, fly at them, and try and get some of those treats. But red birds are completely the opposite. They just want to be left alone. They don't like people. When they hear people come into Central Park, they hide, they flutter and fly away, and they hide in the North Woods. So the behaviors of these birds has actually invalidated your observational data set, making it impossible to answer the very simple question about the relative populations of these birds. And this is not just a hypothetical example. This is happening every day in the hospital records. The behaviors of patients, the behaviors of doctors and nurses and other care practitioners changes what we can observe in the hospital records, making it often invalid or impossible to answer seemingly very simple questions. Examples of these type of things include which diagnosis codes. We can observe diagnosis codes, we can observe lab values and vitals and surgeries, all those data I was showing you before. But we don't know the environmental triggers, the environmental exposures for the patient. We don't know what other hospitals they might have gone to, and we don't know the peculiar treatment decisions or the information that might have gone into that is not captured by those databases. I want to give you a very specific example. There's a series of scores that you can use to evaluate the risk to a patient who has pneumonia. One of them is the CURB, another one's called the FINE score. Now, we've attempted to use these scores by automatically generating these scores from the electronic health record data. In fact, this is a paper published by a colleague of mine that shows that when you apply this score to the electronic health record data to run this automatically, you find something very nonsensical. You find people that are, according to the score, at the lowest risk actually have high mortality. These people should be totally fine. And if you apply the score has been validated. So this doesn't just make any sense. What's happening here is that these patients who died very quickly upon admission or upon arrival, simply don't have very much data in their records. And the fact that they don't have much data makes it look like they're a healthy patient. So these types of biases can totally confound very simple analyses, making the interpretation impossible or very difficult. So data mining is about taking these things into account, about designing automated algorithms that can dive into data sets, account for some of these issues of missingness and bias to generate good hypotheses. And remember, good hypotheses are testable. They make predictions that are testable, and they address these issues of bias and confounding, at least to some extent, so you can do further analysis. And then also, since we have all of these data, we have a new opportunity. Instead of asking one question of one data set, we can ask that same question of totally different data sets using different analytical strategies. And if all of those different strategies tell us the same thing, then that hypothesis is much more likely than others that don't have that same level of evidence.